First of all, thank you very much, Jeremy and Nick, for your very kind invitation. So I slightly modified the title of my talk, and I wish to talk about the T-cell epitope mapping of therapeutic antibodies in healthy donors with the perspective to predict immunogenicity of therapeutic antibodies. Okay, so what is the issue finally with the immunogenicity prediction? We have the molecule, we have the patients, and we expect to know more about the onset of ADA, the frequency, whether they are neutralizing, and for that we try to find tools, predictive tools, to predict these events. And you know that, unfortunately for us, the animal models are considered as relatively low predictive. So we have to find out other tools. And I would say that what we are doing, mainly in many teams, is that we focus on the T cells. And the reason of that is that simply because the T cells are known to initiate the immune response, and they provide help to B lymphocytes. And so when there are no T cells, generally they are very weak response. And you know that the T cells, they recognize peptides presented by HLA class II molecules. So, but what are we doing with the T cell assays? And we introduce as predictive tools the LC donors. And what we wish to do is to get information from the LC donors with the perspective to predict what could happen in the patients. And these patients, it could be ADA patients, positive patients, so they had developed anti-drug antibodies, and others they do not. And you see, there is a real gap between LC donors and patients, in the sense that the T cells are different. In the ADA patients, we expect to find memory T cells that provide help to the B lymphocytes and help to produce the ADA. In the ADA negative, no memory cells, and in the LC donors, we have no memory cells, but naive cells. And the naive cells are really different from the T cell, the memory T cells. As you may know, they are not exactly the cell level of activity. The naive cells are not active, provided they are not activated. But the key point is that there are a really different frequency in the blood. We can find that the naive cells are around 10 to minus 5 or 10 to minus 7. And in the memory T cells, especially for very highly immunogenic viruses, for instance, they could be at very high frequency. So you see, there is a gap, especially because of the frequency. And finally, if we'll investigate what happens in the naive T cells, are we sure that it is really predictive to what happens in the patients? In a sense, the question that we can address is, are we sure that the peptide specificity of the T cells that we found in the naive repertoire really account for what happens in the memory T cells? So this is the kind of question that we can address. So in the frame of the ABIRISK consortium, the ABIRISK project, we are interested in 4 well-known immunogenic antibodies. 2 are chimeric antibodies, rituximab and infliximab. We also work with one immunized form, which is natalizumab, and the well-known fully antibody adalimumab that Jeremy has already presented to you. So we have cohorts of patients which are treated by these therapeutic antibodies, and we have also in the particular work package, we are investigating in the T-cell epitope and of these products. And this is what I will show you today. I will first start with rituximab and infliximab. And in my lab, to investigate the naive cells, what we did is that this is what we call long-term T-cell assays, as we derive T-cell lines specific for the drugs, so the antibody. And this is done by multiple rounds of stimulation in vitro. And then we assess the specificity of the T-cell, either with the drug or with overlapping peptides that encompass the complete sequence of the variable parts. And I will show you some example. With rituximab, for instance, you can see this T-cell line, it reacts with rituximab, not with infliximab. And one peptide pool is positive, and when we look at the individual peptides, you can see a very good response to this peptide. And the same was done also with infliximab. And this has been measured with a panel of donors. We introduced 50 donors, which are HLA types, and we have the diversity of HLA molecules. And you can see that we can get 11 responders from the 15 donors. We find 9 different epitopes that are present on the variable light and AV chains, and there are some peptides that are common to donors. And we have exactly the same picture with infliximab, the same number of epitopes, and also peptides that are common to several donors. As we are academics, I would say we try to really understand what happens with the T-cell epitope, and for this reason, we submit the complete libraries of peptides to HLA class II binding assays. And this is something which is done in my lab. We have a collection of immunopurified HLA DR molecules, DQ or DP, but we just consider DR. These molecules are very common in the Caucasian population. So we submit all the peptides to 11 different binding assays, and this is a very big work, and this can be summarized in only one picture. And if we consider the number of HLA-DR molecules that are bound with one peptide here, so you see that there are many good binders. And in hatch, it's high binders, in blank, the moderate binders. You see that, for instance, here, the peptides, it can bind up to 8 different molecules, sometimes 6 or 5. So there are many good binders. And when we consider the T-cell epitope, you can see that the correlation is not so good. And in fact, there are many good binders that are not necessarily T-cell epitopes, something that we expect. And I already work with vaccine, and this is exactly the kind of things that we can get. But anyway, when we look in more details between the activity of binding of the different T-cell epitopes, so these are only the T-cell epitopes. To make the story short, the values in red correspond to very good binders, and in green is moderate binders. And if we look at the binding activity, you can see, for instance, for this peptide, that it is a very good binder to DR1, and one of the responders to these peptides is also DR1. This can be done with the second responder's donor to this peptide. You see he also responds to DR11, and the haplotype of this and this responder is also HLA DR11. And this can be done with many of the peptides. And I will especially point out one particular peptide in the rituximab is this one. It binds only to DR11, and the 3 donors that respond to it, were also DR11. In other words, what we believe is that most of the T-cell epitope exhibit a good binding affinity to the appropriate HLA molecule. So again, generally affinity play a real role, but is not enough to predict a T-cell epitope. Thanks to the collaboration with Novartis, Novartis is part of the ABIRISK program, we also had access to the mass spectrometry studies, and the idea is that rituximab and fleximab are loaded on dendritic cells. The HLA molecules are purified, the peptides are eluted by ICD treatment, and then sequences by mass spec. And this has been done with 34 different donors, and the results are like this. The peptides are presented by, are retrieved as multiple length variants, having the same HLA binding core like this, for instance, or here. And at the end, we find 5 clustered in the variable part of rituximab in AV chain and 4 in the light chain, and similar data from infliximab. And most of the CD4 T cell epitopes were indeed found by these assays. And I will show you the full picture at the end. Something that you already discussed during this session is the access to patients that have already mounted ADA response. And so we were happy to get these samples and to get the PBMCs of these donors. Unfortunately, there are not so many. So we get PBMCs from 7 donors. One were positive for treated with rituximab, and you see the level of ADA, and the others were treated with infliximab, and we collect these donors from different disease and different services in collaboration with clinicians. And to investigate the responses, these donors, we did not apply the long-term T-cell assays that we use in the naive donors. We only applied the short-term T-cell assays in the sense that the PBMCs of these donors have been cultured with peptide pools during 10 days, and then we submit the amplified T-cells by to IL-5 ELISpots. And what we get as a result is here. For 2 patients, the L and C, we applied the complete set of peptides. I mean, the 45 peptides were introduced in the culture and then submit to the ELISpot assays. And you can see for these donors that there are only 4 epitopes, here, and here, and for the other one, 2. And all these peptides were part of the peptides we identified using the LC donors. And for the others, we only assess whether the peptides we identified are indeed recognized from the ADA, in the ADA positive patients, and it was indeed the case for many, for all of them. We get response to the T-cell epitope we already identify using LC donors. So we conclude that the T-cell response was found in all the donors, focused on few epitopes, and some of these peptides are common to 3 donors. A very brief view of what happens about the phenotypes of the cells. We measure in the supernatant of the ELISpot whether we can find all the cytokines. We did not detect any IL-4, IL-13, which is not, generally very difficult to do, so I'm not completely surprised of that. But we did not detect IL-2. And in contrast, we very easily detect IL-10 in many of the donors, and sometimes also interferon gamma and IL-17. So probably there are a different profile of cytokine response to therapeutic antibodies. Some picture to have a view of what we have done, and you see that from the LC donors in rituximab and infliximab, we have different regions where we can find the T-cell epitopes, and in green, the data we collect from the immunized patients, and you see we have relatively good concordance between the two studies. And if I introduce the data with the binding assays, something funny is that finally we find at least one binding activity for many peptides, you see here. So at the end, if we consider only one binding, it covers almost all the sequence of the variable parts. And if we consider what we call the promiscuous peptides, which means that they bind to multiple HLA-DR molecule, you can see that sometimes they can miss the peptides that we have identified as a T-cell epitope. So it's not really predictive at all. And regarding the maps, you see here we detect very well the cluster of T-cell epitope using the mass spec technology, but sometimes also we have false positive peptides in the sense that they are presented, but they do not stimulate T cells. And the same with rituximab. Just to conclude my talk, I will show you very briefly some data with other antibodies. We also succeed to derive T cells specific for natalizumab and adalimumab, and so you can see here the response for one particular peptide, and it's also here. This is just partial results, and the study is still ongoing, and especially we did not have the data with the binding assays. So just to tell you that with the natalizumab, which maybe it's more difficult to derive T cells with natalizumab with the others, but we succeed to get 6 responders donor. We find 9 different epitopes. And with adalimumab, finally, we have a reduced number of T-cell epitope. And you see that we find only 5. And you can see that one of these epitopes in blue is shared by different donors. And next time I will show you where it is. Okay, just to conclude, so we have investigated the T-cell response to infliximab and rituximab, found the T-cell epitope, and we are happy to see that the T-cell epitope identified in ADA-positive patients are part of the T-cell epitope we identified in HC donors. So it seems that when we consider the naive repertoire, we have good information of what could happen in the patients. Regarding the binding activity, I hope I convinced you that most of the T-cell epitope exhibit a good affinity, but there are also many good binders. And you know, it's a very important issue to know whether we can apply in silico prediction to predict immunogenicity. And clearly, based on this data, we can conclude, I mean, on chimeric antibodies, because, you know, chimeric, the framework parts are from murine origin, that there are too many sites, and at the end, the in silico prediction would be over-predictive. Regarding the naturally processed peptides, so again, we identified T-cell epitopes, and they are part of the naturally processed peptides, but there are also naturally processed peptides that do not elicit response. And we are working on now with natalizumab and adalimumab. I just wish to acknowledge all the people who did the work, and especially in my lab, the two postdocs, Mustafa and Sylvain. So it is mainly in collaboration with Novartis, Sébastien Spindeldrehen and Annette Karl, and people from INSERM in France, who help us to find the ADA positive patients. And the main conclusion maybe is that at the end, it's always difficult to make prediction. Thank you very much.